you can start when you want.
Okay, thank you very much.
Yeah.
I'm compartment. Okay. Everything is fine?
I can see your screen.
Okay, good.
Let us know in the discussion if you have issue with that.
Everything is fine, so.
I think so.
Okay, thank you. So I'm start now. Good morning, everyone. Before starting, I would like to introduce myself. My name is Beatriz, and the Faial Island in Azores is my heartland, where I live until I went to university. I was born in Coimbra, in mainland, city of universities, and my academic career began in 2012. I entered University of Aveiro, where I graduated in biochemistry, and I did master's degree in applied biology in University of Aveiro too. My master thesis is specialized in toxicology, and during my academic career, as well as the beginning of my career, a researcher. I have always worked in parallel in theater and restoration, and in the summer of 2019, I went to work to Association of Producers Native Clams in Fajang da Caldeira de Santo Cristo in São Jorge Island, in Azores too. Between in November 2019 and January 2020, I come to work from IMAR, Institute of Marine Sciences in University of Azores, in a project named Impactor. And today I speak a little bit of this project because I work now in this project again. So, in this conversation, I'm going to talk about my work in the Impactor project, as I said before. It aims to study the cumulative impacts of ocean acidification and copper accumulation in the coral gardens of Azores seabed. I am currently working on the specific task, expression of stress related genes and cold water corals after offshore mining and impact of climate changes. I decided to divide my presentation by colors to facilitate understanding. So I begin to contextualization. Climate change is increasingly evident, and over the years, it will be more and more rapid. In addition to average rise temperature of planet, as well as in sea waters, the concentration of atmospheric CO2 is increasing, and the impacts on the oceans will be huge and still unknown, knowing that the excessive absorption of CO2 by the oceans is leading to decrease in the pH of seawater. This phenomenon is estimated to severely affect seawater's chemistry, including metals and the metalloid speciation, restricting the calcification of the marine organisms and favoring the erosion of habitats created by marine calcifiers, such as species of the cold waters. This scheme show a little bit the ocean acidification. Meaning the extraction of mineral resources from seabed, it's another problem, is the one of the potential most threatening activities from the ocean floor around the world. It will not only destroy benthic habitats, but will promote the dissolution of minerals trapped in the sediment by increasing the concentration of metals such as copper in the water column. It's another big problem because the all ecosystems suffer with this extraction. So copper is a naturally occurring metal that that plays an important role in the cellular oxidation, reduction, redox reactions. And this scheme show a little bit of this. However, excessive amounts of copper in cells cause oxidative stress. For example, it unbalanced redox functions. It promotes the overproduction of oxygen-free radicals. More oxygen-free radicals means more oxidative reaction. So it's the most problem cellular level. I work in this project with two cold waters, Viminella flagellum, it's the white-like octocoral, and Dentomoricea meteor, it's a Sifan octocoral. These two species are native of Azores Sea, and these images catch by the submarine of University of Azores. Okay. So we know the acidification, the ocean acidification impairs the calcification process of corals and decrease the pH and has implications for this physiology. We know that copper toxicity is highly harmful to corals, but we don't know what the iterative effects of the two stressors on coral physiology. These are a first question. It's our first question that triggered this work. How do cold water corals respond to a predictive effect of copper exposure under ocean acidification mixed to climate change scenarios? Several physiological markers are used, like markers of oxidative stress, detoxification, cellular integrity, etc. My work focuses on the evaluation of transcripts. At the moment, I'm analyzing the results in a computer program with the aim of sequencing the transcriptome of these two species of cold water corals, as this was never been done. So I talk a little better later. I will talk a little better later with this thing. Okay, everyone with me? Okay.
Yes.
So this is my experimental drills. So we do 4 experiences, pH normal, pH with copper, pH acid pH, and acid pH with copper too. So the all scenarios, normal scenarios, it's A, and climate change scenario, it's D, for example. During this work, daily monitoring pH and temperature, incubation measurement of oxygen during incubation, and these measurements make the possibility to assess the breathing rates of individual in each of treatments and infer the status of basal metabolism. After 9 days exposure, this is our spread of corals, and it's easier to see in the blue treatment, the coral are health, and purple treatment, the coral are unhealth because of this treatment. When analyzed the biomarkers, we know from the first approach that treatment with copper have different responses compared to treatment without copper. Now we are under computer screening to find out which transcripts are different. It's the most aim of my work. So after exposure, we cryopreservate cold water corals in a liquid nitrogen for RNA extraction. We preservating trisol classic. This RNA extraction, we use trisol. It's the classic method to this work. And when centrifugate and organic phase, right, its reagent is separate from the nucleotides, mRNA. And this is the part to interest me. 3 replies out the 7 per treatment with sequenced. Why these 3? It's not possible to sequence everything for financial reasons. So 3 replies of the initial 7s were chosen random. White, it's a control. White, no, sorry, the blue treatment, it's a control. It's in this treatment bottle with everything the same except the coral. And now I'm do the bioinformatic work. In the last 5-6 years, there has been talk of sequencing the genome of cold water corals with the aim of finding gaps and providing answers to problems that until then no one had addressed, like proteomics, transcriptomics, etc. Some examples of work carried out in this area, but as usual, tropical corals are the most studied and consequently the ones with the most information. There is a little information about the genome of cold waters, gorgonias, which greatly limits to skirting of the transcriptome. Can you explain a little bit of what RNA is and why need the genome? It's the simple scheme. After sequencing the transcriptome of the Gorgonian Viminella exposed to different treatments through global databases and bioinformatics, I have been working on computer program to identify and quantify the genes expressed by corals, exposed to different experimental conditions to understand the behavior of these species when exposed to copper and to change pH. Why the transcriptome? It's a big question. Because the transcriptome gives us information about what is beginning transcribed by the genes at the moment by the cells. Refers to complete sets of transcripts, messenger RNAs, ribosomal RNAs, transfers RNAs, microRNAs, every RNAs almost. They are present in the cell when exposed to an experimental situation. With this information from the transcriptome, we can understand what kind of information genes transmit to cell for translation into function to cell and this translation into functional proteins to respond to Cooper exposure, low pH, and the two stressors. This approach allows us to predict how the Gorgonian Viminella responds to stressors in the climate change scenario. I use this program, bioinformatics solution, to facilitate analysis by RNA sex data. We choose to perform a differential analysis of gene expression between treatments. This type of project is sustainable for analyzing the effects of combined experimental conditions, presence or not of Cooper, and pH change on gene expression. Exactly. Okay. So now I have preliminary results. This program allowed you to explore the transcriptome data in a simple and understandable way. According to the sellers, this software is the program made for biologists, so facilitates the work. For example, this multi-dimensional scaling we see here, multi-dimensional scaling map shows the relative distance between the sequences sampled and allowed to immediately visualize differences and similarities between samples. This is perfect for the first look at differential expression information. It's very preliminary, but you can see different behaviors. So you can trace, yeah, okay, this one. You understand? In the first place, we have samples with Cooper, without Cooper, for example, and this one, it's the samples with Cooper, for example. Bitmap, it's another huge tool, and it's another representation of information are to so-called maps. They show the difference and the similarities of expression values between treatments. With the differential expressed genes, we see the down-regulated ones in green and the up-regulated ones in red. Yeah, this. First heat map represents this first heat map represent all genes. However, for a better interpretation of data, we select the top 50 genes. It's the most frequent genes. It map to get a clear overview of data and as starting point for interaction and analysis. This shows the relative genes expression levels in RNA-seq segment, being able to assess the cold water corals resilience to change in pH and the presence of absence of Cooper. So we don't have conclusions now because this work, I do this work now yet, but the preliminary data point to transcriptional response of cold water corals to decrease pH and Cooper exposure. With this work, it will be possible to identify genetic biomarkers and physiological stress to Cooper exposure for low pH exposure and for exposure to both. And the second one, identify basal genes that respond to experimental conditions, being species of deep habitat. Acclimatization to Aquarius causes some physiological causes some physiological changes. So I think it's that. Thank you for listening to me these minutes. I still at the beginning, both in my career and in this part of the job, but I increasingly believe that the key to success is the teamwork. So thank you very much. Some questions I'm allowed.
Thank you, Beatriz.
Okay.
Really nice talk.
Thank you. I stop screen yet? No.
Yeah, you can stop sharing the
okay.
screen.
Okay, thank you.
So yeah, if anyone has a question, you can just raise his hand and speak, or send it to the comment section in the discussion. So yeah, feel free to ask question to Beatriz. If no one has a question for the moment, I have one first. You show in your experimental design that you have choose pH 8.1 for the normal condition. I guess that's the basal pH. Yeah, and 7.8 for the acidification.
Yes.
Do you know what are the prediction for pH decrease for ocean acidification? Like when is it expected to reach this pH? This 7.8, and is it expected to go? I guess it's expected to go even lower if it goes.
It's the one scenario, but yeah, we expected the pH low to 7.8 in 2050.
Okay,
and the temperatures low too. Up. So in the bottom of ocean of Azores, it's 4 degrees now, more or less, and we expected will go up to 6.
Right. So I guess you will also like to test that at some point, like not maybe in your work, but test the temperature effect.
Yeah. No, because in this work I don't work with the temperature. It's so I control the temperature, but I don't work with this stressor.
Right. We have one question from Monty P in the comment. He's asking, what is the expected source of copper in this environment? Why are you testing copper?
We have this work in a laboratory, and we have all experimental control, and in Azores we don't do mining, but we study this possible the impact, because the political decisions are based in these studies. In Azores works this way.
Okay, and yeah, maybe it's also because like they are living close to the ridge system, where they are planning to do deep sea mining there. I guess it's because of that. We have another question from Catherine Murray, and she's asking, how was the concentration of copper using the experiment chosen?
My PI has an article, scientific article, Inês Martin et al. 2018, and he studied this effect LC 50. Is the first lethal toxicity study of copper. So in this study, she proved this concentration are good to do this type of works. Can I put the reference in chat if you want.
Yeah, of course. Feel
Okay.
free.
Because this article explains everything. The reason of we choose this concentration.
Okay, thanks. Yeah, well, does anyone else have a last question, maybe? You will also have time, as I said, to ask them at the end after Emilie's talk.
Okay.
And
yeah.
question.
Okay. This is a reference. Okay. Thank you very much.
Thank you, Beatriz. So we're going to start now with our second speaker, Emilie Chagny. We're going to talk about the peptidases in archaea and their role in their adaptation to the deep sea environment and the biotechnological interest of these peptidases. So, Emilie, you can go.
Okay. Okay. Is everything okay? Can see my screen?
I can see it.
Okay.
If anyone has trouble, just let us know in the chat.
Okay, so hello everyone. I'm Emilie, and today I was given the opportunity to present you my PhD project, which I started in October. And so it aims at studying new peptidases, their role in the adaptation to deep environments, and also their potential biotechnological applications. So this work is conducted within the BIP research unit, which is part of the Ifremer Institute in Brittany. So BIP stands for Biology and Ecology of Deep Sea Ecosystems. And there are four main research topics. So there's the biodiversity of deep sea ecosystems, the interactions in those ecosystems, the responses and adaptations of organisms living there. And there's also some work on methods and tool development to study those environments. And so my work is part of the third research topic, and I will be especially interested in adaptations in deep sea archaea. So when studying thermococcales, which are hyperthermophilic deep sea archaea, four very interesting peptidases were found. So they caught attention because they have a very special shape, but I will talk about that a bit more later. And they are also more specialized than other peptidases. And very interestingly, they have complementary activities. So when combined, they can degrade virtually any peptide. And so it has been hypothesized that those peptidases have a link with the adaptation of thermococcales to their extreme environment. So just to lay some grounds, I will give a quick definition of a peptidase. So a peptidase is an enzyme that will degrade peptides, so polymers of amino acids. And so here we'll be interested in exopeptidases, which are peptidases that degrade the final amino acids of the chain. And so my work focuses on TET peptidases, which are named after their shape, because as you can see, they form a tetrahedral complex made of 12 subunits. And so they are believed to play a role in the central metabolism of the cells. And so that's why we think that maybe they could play a role in the adaptation of a species to the deep environment. And so in my project, I will be studying those peptidases following three main axes. So first, there will be a focus on their potential role in the adaptation to deep environments. And so here, when we talk about deep environments, we're talking about the adult zone, which is everything situated below 6,000 meters in the ocean. Those depths mainly occur at subduction areas, so in subduction trenches. This is an extreme environment because at those depths, there's no more light, the pressure is high, the temperatures are low, so typically around 2 degrees. But surprisingly, there's a very high microbial activity down there, and there's a lot of biomass. So this can be explained by what's called a funneling effect. So because of the V shape of the trench, the organic matter that's produced at the surface will fall and eventually deposit at the bottom of the trench. So there will be an accumulation of organic matter, but this matter has a particular composition because it will have been degraded by organisms during its fall. And so when it reaches the bottom, it will be enriched in so-called refractory compounds, which are harder to hydrolyze. And so here we'll be asking if TET peptidases maybe allow the degradation of this refractory organic matter. Then we'll also be interested in those peptidases from molecular phylogeny point of view. So those peptidases are not found in all archaea. And when archaea have them, they can have between 1 and 4 of those peptidases. Preliminary studies also showed that some of those peptidases have an archaeal origin, but some would have been inherited from bacteria through horizontal gene transfers. So here we'll be trying to reconstruct the evolutionary history of TETs to understand how we came to this distribution of peptidases in archaea. And so finally, we'll also be interested in potential industrial and biotechnological applications of those peptidases. So as I said, those peptidases have they have a an interesting specificity of substrate. Heterocomplexes have also been observed. So this means that it would be possible to combine different substrate specificities and different activities in the same complex. Those enzymes could also be immobilized, and once formed, the complex is very robust. So this means that it could resist to the harsh conditions used in industrial processes. And so for all these reasons, TET peptidases seem like good candidates for industrial applications, and for instance, in the agri-food area. So this project will be divided in 4 main steps. The first one being to search for new TET peptidases. So to do so, we used identification criteria. And using those criteria, we searched in public databases of archaeal genomes and also in mags that were constructed at the lab from deep sea sediment samples. So those are the results for the search in public genomes. So it allowed to retrieve 2,111 sequences. And so as you can see, 7 groups emerge on a tree. So the sequences were placed on a tree, and we could see 7 groups. So there are the TET 1, 2, 3, and 4 groups that I already mentioned, and that are the 4 peptidases we can find in Thermococcales. But then there are also new groups, so the TET 5 here and TET 6 here, that are only found in allophilic species of Archaea. And then there's this big category over here. So it's the rest of them. And so it's called TET others. And there's more than 1,000 sequences in this big category. Then we were interested in the distribution of those groups in Archaea. So as I said, TET 1, 2, 3, and 4 were found in Thermococcal species, but also in some other species, as you can see here, and so here's the second part. So in some Asgard sequences in here. The TET 5 and TET 6 groups were only found in Allobacteriales, so allophilic species. And then there's the TET other category, which is the more widespread. As you can see, it's found in a wide variety of organisms. And then there are also species, where no TET peptidases were found, for instance, here. And I indicated by red stars the sequences that were found in the MAGs from the lab, so from adult samples. And so those results were consistent with what has been observed in public genomes. So then we had to choose, let's say, the most interesting sequences to try to better characterize them. So the aim here was really to explore the diversity of peptidases, of TET peptidases in Archaea. So to do so, we went back to the tree again. So TET 1, 2, 3, and 4 were already characterized and studied, and so we decided to leave them aside. And we focused our efforts on TET 5, TET 6, and TET others groups. So as I mentioned, TET 5 and 6 are only found in allophilic species. And proteins from allophilic species can be fastidious to purify. So we were very interested in Allophyrax volcani sequences because this is a moderate allophile. So this means that it does not require as much salts as other species. So those proteins could be easier to purify. It's also a model organism, so it was quite convenient. And even more interestingly, it had two TET peptidases, one of the TET 5 and one of the TET 6 groups. So we chose those two sequences as representatives of those groups. And so then we were left with this big category, but some sequences caught our attention. So there are sequences of the Asgard group that presented a very particular insertion, as you can see here, of about 20 residues. It had a very strong charges contrast. So here you have positive charges next to negative charges and a lot of charges at the same site. And so those are all sequences from the Asgard group, which is also very interesting from an evolutionary point of view, because it's the closest lineage to eukaryotic cells. So we decided to go for sequences that had this particular insertion. And so we chose 4 Asgard sequences. So we chose 2 of an Heimdall Archaea because this genome was reconstructed from samples from hydrothermal vents. So those proteins could be assumed to be thermostable, which is also nice for their purification. So we got 2, one with and one without the insertion I was talking about. Then we chose a peptidase from Prometheoarchaeum synthrophicum because it's the only Asgard Archaea that has been cultivated in a lab for now. So it seemed very interesting too. And then we chose one sequence that came from the genomes reconstructed at the lab, so from adult samples. And so this is from Thor Archaea, and it also has the insertion. So those are the 6 sequences that were selected until now. And I started, so we ordered synthetic genes that were cloned in a plasmid to allow their expression in E. coli cells. And I did some tests for the 4 Asgard sequences. So I will present you the first results. So this is what the tests look like. So here we're expecting a protein of about 40 kilodalton, sorry. So that would be the band you're seeing here. Then we want to get to see if the protein is expressed. So here, as we have bands, it is expressed. Then we want to see if the protein is soluble. So here you have the supernatants of the samples here and here. So for S, and then you have the pellets, which are the P. And so soluble proteins will be in the supernatant. So as you can see here, we have bands. So this protein is soluble. And then we did a heat shock to see if the protein is thermostable. And so as the bands are still here after the heat shock, it means that this protein is thermostable. So I did some tests for all 4 of them. And we tried different conditions for the expression and different temperatures. And so, as you can see, we got conditions where 3 of them are soluble. So that's nice. Out of the 3, 2 are thermostable. And we'll still have some work to do for this one because for now it's not soluble. So it will be hard to work with. So yeah, as I said, we'll still have some work to do to optimize the expression and solubility of the proteins. And I will also have to do the same work with the 2 allophilic proteins. Then we'll have to purify those proteins. Once we'll have purified proteins, we'll be able to move on to the characterization of those proteins. So first, functional characterization. So we'll do degradation experiments to determine their substrate specificities. And so this will allow us to conclude on their potential role in the adaptation to deep environments, and also to maybe find some biotechnological applications from them. And then we'll also be interested in characterizing their shapes, their structure. And so this will be done at the Structural Biology Institute in Grenoble. And yeah, so that were my future perspectives. And so there's a few people I would like to thank. So first of all, thank you for listening to my talk. Thanks to the DSBS for hosting this talk. Thanks to Didier and Bruno for supervising my work. We also got some help on the bioinformatic stuff from Simonetta Gribaldo and Najwa Taib, and we'll have some help on the characterization with Philippe Soudon. And also, yeah, thanks to the people who financed this work, sorry, and I'm done.
Thank you, Emilie. That was a very nice talk.
I will be happy to answer any question.
Yeah, thank you. So yeah, feel free to ask questions if you have some. If no one have one yet, and take a bit more time, I have just one question. Mm-hmm. So you mentioned that in several species you can have like between 1 and 4 TET peptides, right? Have you seen, or do you expect a link between the fact of having several TET peptides and some specific environmental context or factor, like for example, like the fact of having like the species that have more peptides, they have a wider depth range, or they are in the lower depth, or other factor maybe?
So for now, the maximum, so 4 peptidases, this was only found in thermococcal species. And so we know that these are heterotrophic species. And so this could be linked to their metabolism. Then we found 2 or 3 in the allophilic species. And it seems like other only have 1. And there's also this thing from whether it's archaeal or if it has a bacterial origin, because the hypothesis would be that maybe they all had the archaeal peptidases at the start, and then some of them got bacterial tests, so the Thermococcales, for instance, and maybe this was more efficient, and so they lost the archaeal TET peptidases, which would be more generalist, but so maybe less efficient. So it's more about maybe their metabolism and whether they're heterotrophic species or not. But for now, that's something we would like to gain some insight on. But yeah, those are perspectives.
Okay, cool. Thanks. Yeah, maybe you can. I don't know, and maybe it's beyond the scope of your work, but maybe you can test that with like maybe not all the data set, like the of genome you found in the database, but some selected one where you have a bit of background in their environmental context or their specific metabolism or physiological condition, and yeah,
that's also something that's tricky because we are working with metagenomes and MAGs, and so there are species where we just don't have informations, and so yeah, that's also a hard part of the thing.
Okay. We have a question from Frank Lesrovsky on the chat. He's asking, is it possible to predict whether TET peptidase has specific peptide targets?
Actually, no, because the sequence is really conserved among all of them. And it's not possible to like predict their activity based on their secondary sequence. And so the specificity of the peptidase really comes from its oligomerization and from the complex with the 12 subunits, because it assembles, so first into dimers, then hexamers, and then the dodecamer. And in the complex, there are like catalytic chambers, and it's really the shape that gives the specificity and the environment of those catalytic chambers. And so that's actually also a hard part of it, because when we wanted to select sequences, we could not say, oh, this one looks like it's going to degrade something. We didn't know. And so that's also why we choose to select our sequences to like explore diversity of archaea and not to target one substrate specificity, because we could not predict it.
Thanks.
Thank you.
So does anyone else have questions for Emilie? Maybe not yet. Yeah, so if you think about something later, remember you can ask them directly to the speaker. You can use Slack, the DSBS Slack. And yeah, if we don't have more questions, I think we can end the talk for today. So yeah, thank you all for joining.
Thank you.
Just one message from the society. You probably are not concerned if you are here now, but we have quite a decrease of membership recently, probably because we don't have a symposium this year. But yeah, if you don't have renew your membership, and you want to continue to enjoy the talk of the society, the seminar series and everything, don't hesitate to go on the website. You can, if you don't know how to do it, just let us know and we'll help you. And also one last announcement. Since we don't have symposium this year, we will do our annual general meeting, the AGM, during the Challenger conference, but we will do that online so everyone can attend. And we will let you know about the detail of that later with the email. So thank you all for joining today.
Thank you for
see you in, of course. And see you in September for the next one.
Thank you.
Bye-bye.
Bye.
Bye.